Hi there, and welcome back to the channel. I'm the GCSE science teacher, and in today's lesson, we'll be learning about culturing microorganisms. This is for students who are studying the triple science or single science biology exam. If you are a higher combined or foundation combined student, you won't need to know this for your particular exam. If you're also an A level student for biology, this video will be very helpful as a bit of a recap too. Content today includes microbiology, aseptic techniques, method writing, interpreting the data, colony calculations, population growth calculations, and also if you're a higher tier student, you'll need to know how to express your answer in standard form. So let's get started. What is actually microbiology? It's a subcategory of biology where scientists will study and research small living things or microorganisms using microscopes and also other types of equipment such as agar gels, which we're going to talk about today. One thing you need to be aware of for your exam is that bacteria divide through a process called binary fission. This is a very fast, rapid type of cell division, which is not like mitosis or meiosis. And I'll cover those two cell division types in another video. Often, bacteria, particularly, can grow quite quickly. Around every 20 minutes, they can divide. And this is particularly helpful if they have specific nutrients or temperatures that they are working optimally at. And the reason I say it's helpful is because we as scientists can use those bacteria to actually test to see the efficacy or how well a product works, whether it's an antibiotics that we want to check how well that actually treats bacteria or bacterial diseases, or it could even be disinfectants, and we want to see how well that disinfectant or that chemical actually works against that bacteria. So, as we grow bacteria, we need to make sure that we only grow the particular bacteria we want. And that way, we can actually ensure that our results are accurate. Also, however, we don't want to have bacteria just lying around and could potentially cause harm and contaminate other parts of the lab or all the parts of the equipment that we have. So, we actually exercise something called aseptic techniques. And this is to prevent that contamination because if we start to culture bacteria that we don't want, they can actually outcompete the bacteria that we do want for nutrients and also space. One of the ways we ensure aseptic techniques has been abided by is by washing our hands with antiseptic soap. We also work very closely to a Bunsen burner flame. That way, the air surrounding our agar gels is not contaminated or have any airborne bacteria within the air there. Also, we want to sterilize any equipment with the Bunsen burner flame, and this equipment is often made of metal, so we can pass the metal through the flame quite nicely. And we also want to use any sort of sterilizing solution, whether that's a Virkon or any other kind of chemical. Other ways we can ensure aseptic techniques have been abided by is by using cultures and petri dishes or agar gels, which actually have been sterilized before use. And after we use those plates, we actually want to destroy them, and we can use an autoclave machine to do that. Essentially, it brings the bacteria up to incredibly high temperatures and just kills them off. So, in the exam, you'll be given potentially one or two different types of methods to write about or to answer questions about. And one of the methods I like to share with my students is when you are writing a method, you want to ensure that you are giving enough details. So, if someone else was to do that method just by using what you'd written, they could actually get results which are repeatable and actually make logical sense as well. So, one of the ways to do that is when you're writing your method, you want to ensure you're using bullet points to structure your answer. You also want to ensure you're using correct names of equipment when you write your answer too. And that way, your examiner will be able to see actually, has there been logical thought and a logical flow in your answer? Because what will happen is it's usually worth about 6 marks, and your examiner is going to mark it based on the logical reasoning in your answer. This is what we call a leveled answer. So if you look at mark schemes, you'll see you've got level one, level two, and level three. And level three is essentially 4 to 6 marks. That is how you're going to get all of the marks by writing it in a logical way. So let's go through the method. The first thing you want to do is spray the work surface that you're going to be using with a disinfectant and wash your hands with an antibacterial soap and turn the Bunsen burner on. You want to be using a flame that's a roaring flame, so that the area surrounding your equipment, the air has been cleared and destroyed of any bacteria that may be there, and that way nothing will fall into your agar gels. You also want to be wearing goggles during this point, by the way, because it's going to be we're going to be using chemicals, we're going to be using bacteria, and you don't want to get anything in your eye, so goggles are a must. The second thing you want to do, your technician or your teachers will often do this for you. So if you've done this in class, this has probably been prepared for you to save some time in the lesson. But essentially, you want to prepare and set your sterilized agar gel overnight with the bacteria broth incorporated. So the gel itself will have the bacteria already in there. And we call this a lawn. You can also get different types of agar where you can make streaks on the plate to check the colonies and see how well they grow. But often this one is done so you can actually test the efficacy of antibiotics or antiseptics as well. So you have your agar gel. This is usually the bit that you get when you're in class, and you want to label the agar gel carefully, not the lid, because the lid can be removed. You want to label the base of the gel with your name or initials, the type of bacteria species you're using, maybe it's an E. coli, and you want to write the date as well. Okay. The next thing you want to do is you want to draw on that plate three different lines. So you're separating the plate into three equal sections. And this is so you can place filter paper discs that have been soaked in an antiseptic or an antibiotic, and you can place those into those three different sections. Because what we're doing in this experiment, we're actually testing to see how well those, whether it's an antiseptic or an antibiotic, how well it actually works at destroying and killing and stopping the growth of that bacteria. And to do this, you want to open the lid carefully, open it only slightly so you can basically put the disc in, but you're not opening all the way, and that's just to prevent any bacteria or contaminants in the air getting into the gel. You also want to work very closely to the Bunsen burner flame, and you want to use a pair of forceps, which are metal tweezers, and you want to run that through the flame first before you pick up any of the filter discs. After you've done that and you've placed each of the filter paper discs that have been soaked in the antibiotic or antiseptic into the correct area, you want to tape down the lid to the base of the actual agar gel. And you want to make sure that on the plate itself, you want to label what had actually gone in each of those, so you know what antiseptic went where. The reason also for the tape is just to ensure that the actual lid stays secure, but make sure you don't tape all the way around. That way, you would actually prevent oxygen getting into the agar gel. And if there's no oxygen there, you actually would encourage very harmful pathogenic bacteria to potentially grow, and that would be very dangerous. So you need a little bit of airflow through there. So once you've done this, you can then incubate your gel at around 25 degrees or lower for around 48 hours. And it's really important that that gel, that agar, doesn't get a higher temperature than 25 degrees, because any higher than that, we're going into human body temperature of around 37, and that can encourage really harmful pathogens to grow, or microorganisms that cause disease, and we don't want that. So we only keep it at 25 or lower. Once that's done, you will actually have wonderful plates, hopefully, that show lots of results of zones of inhibition. We're going to talk about that now. So let's imagine we've fast forwarded in time and we're going to interpret our results. So you'll get a plate and it will look something like this, which let me talk you through it. So we've got our three sections, A, B, and C, and you can see we've got circular discs called zones of inhibition. And these are areas where bacteria has been destroyed or killed or prevented from growing. The bigger the zone, the larger the area, the better that antibiotics has worked at destroying the pathogen, or the better that antiseptic has worked. And we can actually calculate the area quite nicely. So let me show you how to do that. We want to measure the radius or the diameter with a ruler, and we want to take a couple of readings and calculate the mean average of that. So you can see I've done that here. We've got 12 millimeters and 15 millimeters. That's the diameter. Divide those by 2, we get the radius, and we can pop that into the area equals pi r squared equation. Don't forget to square, and you should get an answer of something like this: 143.1 millimeters squared. And we can compare. I mean, looking at it objectively, you can see that uh, the circle that I've calculated is actually the biggest zone, just like by looking at it. But we can actually quantify that and show that as a proper result. So once you've done this, you also might be asked in the exam to calculate population growth. So let's run through this now. We have an example. It says the mean division time for a bacteria population is 20 minutes. If the observations begin with one bacterium, calculate how many bacteria will be present after 7 hours. So we're trying to calculate how many times or how much divisions we will get. So first thing you want to do is calculate how many times the bacteria will divide in 7 hours. So we know that the division time for this particular bacteria is 20 minutes. So in an hour or 60 minutes, if we divide that by 20, we can say there are 3 divisions per hour. We then want to know, in the question it says how many bacteria will be present after 7 hours. So we will do 3 times 7, which is 21. So there are 21 divisions in 7 hours. We then need to calculate the number of bacteria in the population. So we use the number of bacteria at the beginning, which is just 1, and the number of divisions, which is 21 in 7 hours. And we put this into a calculation of 1 times 2 to the power of 21, and we get an answer of 2.09 times 10 to the power of 6. So you can see just from one type of bacteria, it will divide 21 times within 7 hours, and you'll get a colony of 2.09 times 10 to the power of 6. So it's exponential growth. It will divide and divide and divide very, very rapidly indeed. Now, you might be wondering, if you're a high tier student, how do you do standard form? Now, this is a whistle stop tour of how to do this. I'm more than happy to go through this in another video if you wish. But one of the best ways to understand this is to start off simply. So 10 is the same as saying 10 to the power of 1, okay, because there's 1 0. If we do 100, we've got 10 to the power of 2, 1,000, 10 to the power of 3, 10,000, 10 to the power of 4, and so on. Okay, so it's just a way of writing out a very large number, but also a very, very small number in a nice, concise way. Because often in the previous slide, you saw that I had a big number and it just looked very messy, and you don't obviously want to have to write out that full number every time, especially if you're a scientist and you're writing out reports. It just makes the numbers look better and easier to work with. So another example of this, we could say 4 times 10 to the power of 3 is the same as saying 4,000. One of the other things that students often find quite tricky is when you're going into decimals. So let's go through that now. 0.002 is the same as saying 2 times 10 to the power of minus 3. And I'm getting that information because if you look at my decimal point there, if we move it to the right, we go 1, 2, 3, we can actually get the 2 back to where it is, and that will be going the opposite direction, so a minus 3. And then another example I'll do here, we've got 2925 245290, big number like that. Essentially what we're doing here, this is a really big number, we can actually reduce this down to 2.92 times 10 to the power of 6. Essentially, I have just counted how many points away from the decimal that this number would have, and I've just reduced it down. Often in the exam, they will give you extra marks for knowing how to do standard form. If you need some more help, let me know, and I'm more than happy to do that. Like I say, thank you so much for watching. I hope that has been helpful to you. I've been the GCSE science teacher and you have been curious. If you did enjoy, please feel free to like, comment, subscribe, and share this video with anyone you think would find it helpful. And in the meantime, if you would like more GCSE content, you can always check out my channel for the videos I've done at the moment. I'll be posting more regularly, at least once a week, so you'll see me on there, but also on my Instagram for bite-sized revision at the GCSE science teacher. And until next time, have a great day and I'll see you in the next one. Bye.